We're starting with a cell that's in G1 of interphase. That's the beginning of interphase. And in this organism, there are two different kinds of chromosomes. The long chromosomes, which are red and yellow, and the short chromosomes, which are blue and green. The red chromosome and the yellow chromosome are homologous, meaning they have the same genes in the same order, but might have different versions of those genes, big A versus little a. During S phase of interphase, DNA replication occurs. Now there's twice as much DNA in the cell, so we have 4 copies of every gene, 2 copies of the big A gene and 2 copies of the little a gene, and so on and so forth. But the number of chromosomes is the same. There are still 4 chromosomes in this cell. Before mitosis begins, both DNA replication during S phase and centrosome replication during G2 must occur. At the beginning of mitosis, the nuclear envelope begins to break apart, DNA condenses, and the centrosomes begin to migrate to opposite sides of the cell. During prometaphase, the mitotic spindle begins to attach to the chromosomes via the kinetochore proteins as the nuclear envelope finishes breaking apart. At metaphase, the microtubules have pushed and pulled the chromosomes to line them up along the metaphase plate. This is an imaginary plane across the center of the cell. During anaphase, the kinetochore microtubules shorten and pull the identical copies of DNA, or sister chromatids, further apart from one another. At telophase, the mitotic spindle falls apart, the nuclear envelope begins to reform, and the DNA decondenses. Meanwhile, a contractile ring is forming between the 2 cells to pinch them apart. The place where they're pinched apart is called the cleavage furrow.